Hi, everyone. Welcome to episode 290 of our Tick Bootcamp podcast. The title of today's interview is Reductionist Cure, an interview with Dr. Janis Weis. My name is Matt Sabatello.
My name is Richard Johannesson.
We're honored to share the knowledge and wisdom of Dr. Janis Weis. Her research areas of interest are immunology, microbial pathogenesis, genetic regulation of Lyme disease severity, and pathogenesis of Borrelia Burgdorferi. Lyme disease is the focus of her research, and she's made some fascinating discoveries that can help real-world patients like you and I. Her next steps are to take what she's learned at the research level and bring it to the clinical level and start working with doctors and patients to apply the lessons she's learned in the lab to help us feel better.
Hello, Professor Weis, and welcome to the Tick Bootcamp podcast.
Hello, I'm glad to be here.
Well, thank you for getting up so early and looking so wonderful at 7 o'clock in the morning your time. And what we like to do on our podcast, Professor Weis, is we like to give our folks a context for our guests. So why don't we first talk about where you're currently working?
I work at the University of Utah, the School of Medicine. I'm in the Department of Pathology, and it's in Salt Lake City.
And talk to us generally what you do there and give us some sense of what the most enjoyable elements of your job are, including the research and the work that you're doing with your students.
So most enjoyable part of being an academic professor for me is getting to work with graduate students, getting to think about doing research on an important problem and working in the lab on that problem, but also having students that are excited and energetic and knowledgeable who want to come and work in the lab with me. And we have lab meetings and we exchange ideas and work on problems together. So for me, it's working in the lab and it's working with students is the most enjoyable part.
So, and it's not just your students that you've worked with. I understand that you have actually a couple of children who are working in the family line. So talk to us about your family and give us some background on where you went to school.
So I grew up in Kansas City, and so I did my undergraduate degree in microbiology at the University of Kansas in Lawrence. So I'm a Jayhawk.
All right. Go Jayhawks, great basketball school.
Go Jayhawks, right. And then after I graduated, I went to the University of Minnesota in Minneapolis, and I did a Ph.D. in microbiology. I worked with Pat Cleary, who is a wonderful group geneticist working on group A strep, which is the bacteria that causes strep throat. And it was in Minnesota that I met my husband, John Weis, and we married in Minnesota, and then we took the next step for us was to move to Boston, where we were post-doctoral fellows at Harvard Medical School and Brigham and Women's Hospital, which are affiliated.
So I understand you and your husband had common interests when you were both graduate students, and then what did your fellowship. So talk to us about what type of work your husband was doing at the time that you had met. Talk to us about the fellowship that you did together, and talk to us about the type of work your husband did before he passed.
So my husband was a virologist at the University of Minnesota, also in the Department of Microbiology. So he worked with Tony Ferris and worked on understanding assembly of a Rouse sarcoma virus. And then we moved to Boston, and he did a postdoc with John Seidman on immunology, molecular immunology, and I did a postdoctoral fellowship with Doug Fearon on complement, which is a complex pathway of serum proteins. It's very important as a defense against microbial infection and also against autoimmunity. So it's yeah.
So the two of you had these parallel paths that you're on academically, and now, of course, you were married, and then the two of you came out to the same university to work. So talk to us about what it was like to have this sort of partner in life and partner in first studies, and then ultimately in the research work or at least research that you were both doing at the same university.
So when we were still in Boston, we had our daughters, twin girls, and so that sort of made life a little bit more complicated. Boston's a really fun place to live, but it's congested, and we were ready to leave and establish our own labs. And so we moved, and we were very fortunate. The University of Utah had offered us both positions, tenure track faculty positions. So we moved to Utah. John and I both had separate labs, and but we had our joint lab meetings and really helped each other out with our students and postdocs and interactions on campus. So his lab was more fundamental processes of immunology, and I, just before leaving Boston, I met Sam Telford and Jose Riviero, who were two very outstanding tick biologists, and they taught me how to work with the Lyme disease spirochete, Borrelia Burgdorferi. And so so I had just taken on that project when we moved to Utah, and I, as a microbiologist by training, I had really missed working with the bacteria. And the first time that Sam Telford showed me a Borrelia Burgdorferi swimming under a dye-filled microscope, and I saw these wiggling spirochetes, and it reminded me of something from Indiana Jones in a snake pit. I was hooked, and I said, this is where I want to work. This is the project where I want to go with my career. I want to take the things I learned as a postdoctoral fellow, return to microbiology. And Lyme disease was an emerging pathogen, an emerging disease, and there was opportunity to bring expertise from different fields into into a study and really make progress, because it was relatively new, and new information is coming out. So
let's pause there for 1 second, Professor Weis, because you have two Lyme geeks on here who are very excited to talk with another Lyme geek, and it was really cool for you to outline for us how your passion for becoming a Lyme geek was triggered. But let me build out one more piece of your personal story before we move forward, and that is, talk to us about your daughters, because of course you would wonder what two PhDs would ultimately produce, right? So the two of you are on these these geeky academic tracks. You're both doing fellowships at Harvard or doing postdoctoral research at Harvard. So what is produced when two twins come to two geeks who are doing their work at the top university in the U.S.? What talk to us about you two daughters and what they're both doing today.
So once we moved to Utah, we stayed in Utah. And our daughters were definitely interested in going into the lab with us, and seeing what we did in the lab, and coming in, and a Saturday morning in the lab was a fun thing to do. Dinner table topics were probably different than the average dinner table. I remember when our daughters were in high school and a friend was coming over for dinner, it's like, Mom, don't talk about your work at dinner. Talk about something else. Don't talk about science at dinner. And So, and when they were in in grade school, they did a science project, obviously helped by us, where we were titled "Do Identical Twins Have Identical DNA?" And so we did some genome mapping, very, very couple of genes, but just to show that they were similar in our daughters. So we were kind of geeky, I would say, pretty geeky. But we did, one of the nice things about living in Utah is you can be outside, and we spent lots of time outdoors. We were really close to the Grand Tetons and Yellowstone, so we took a lot of trips there, and they love nature, and and just playing outside. And twins are the same age, and so they can get out and play with each other. And so we did a lot of hiking, biking, skiing, camping, things like that as they were growing up.
All right, well, so you're unfortunately begging for me to ask the question that I have to ask as a Lyme researcher who has two young children who are now enjoying the outdoors. Tell me, what kinds of things were you doing as a mom that were designed to keep your children safe from coming in contact with ticks and tick diseases?
Right. So, you know, the interesting thing is in Salt Lake City, we don't have many ticks, and the Ixodes ticks are not common. In fact, they're rarely identified in Utah. The environment is just too dry for the ticks, and so it hasn't, for us, this incredible need to constantly be screening for ticks is not as big a problem in Utah or the Tetons or Yellowstone as it is on the East Coast. So we took regular precautions of just using insect repellent more for mosquitoes and other kinds of insects, but in general, we weren't very concerned about tick-borne diseases in this area.
So before we come back to discussing what your daughters are doing today, I do want to build out that piece of ticks and tick biology. When we interviewed Dr. Thomas Mather from the University of Rhode Island, one of the things he talked to us about was what ticks need to survive, right? And he said they need blood and they need moisture, right? And that was one of the tools, and that is one of the tools that we encourage our community to be aware of when they're interacting with nature, because you're more likely to come in contact with ticks if you're in a place where there's moisture. And obviously, you know, because ticks will need blood meals, they will, of course, the more animals that we come in contact with, the more likely it is that they're going to be infested with ticks. So in Utah, because the climate is so dry, there is not adequate moisture for the ticks. Is that the reason why there aren't many ticks in Utah?
That's my understanding. I'm not a tick biologist, but that is definitely what I have been told. It's not that there's anything that's preventing ticks from coming to Utah. It's just that those that do come in on birds, etc., don't find the right environment.
Right. Okay. So let's finish this piece of our conversation. So talk to us about what your two daughters are doing today. You have these identical twins, and you said they are identical twins, right? You have identical twins who are being raised by two geeky PhDs. And I'm assuming, you know, they perhaps put limits on you the way my children put limits on me. I'm not allowed to talk about Lyme disease or ticks for any more than 10 minutes anytime we have company at our house, because my children place limits on me. So I'm assuming you have the same limits placed on you by your children. But talk to us about where this childhood experience took your two daughters.
So when the girls graduated from high school, they wanted to go to small liberal arts colleges. They wanted to leave Utah, and they wanted to go to a college that neither of their parents was a faculty member. In other words, even though it would have been nice for them to have stayed in Salt Lake City and taken advantage of tuition benefits at the University of Utah, they really needed to leave. They needed to leave being in the shadow of my husband and I, and they needed separation from each other. So my daughter Julie went to the University of Puget Sound, and my daughter Allison went to Lewis and Clark. So they went to the Pacific Northwest small liberal arts colleges and explored different types of degrees. But Julie ended up with a degree in molecular biology, that's right, and Allie with a biology degree. And then they both, though, took time deciding what they wanted to do if they wanted to proceed to do advanced degrees. They just needed time for some life experiences. And then eventually, so they did some traveling and working outdoors, and then kind of came back to lab type of things. And Allison ended up getting a PhD in microbiology at University of California in Davis. And Julie ended up going to medical school. She did a master's of public health in Seattle, but she came back to the University of Utah for medical school, a large reason because my husband was ill and she lived with us for his last few months of life, and she did her medical school degree here.
Congratulations! So it's you should be a very proud parent, and they're doing. It's wonderful that they've stayed in the family trade, if you will.
Well, we try to encourage law or something where they do have good arguing skills, it seems like. But I mean, that's probably a bad assumption on my part, and you'll tell me.
No, it's a good assumption, but of course they were more passionate about getting into the lab than they were with developing the argument skills that they had to develop to keep their mom from talking too much about ticks and tick diseases, which is one of the reasons why I think I have a couple of lawyers, because they're constantly reining me in and preventing me from talking about ticks and tick diseases. So in the spirit of moving back, so talk to us about the work that you started to do when you came to the University of Utah, and how this passion that was built into you when you were in the Lyme Belt in you know, you were doing your research and your fellowship at Harvard here in Massachusetts. You brought, you know, it's sort of like you got this passion. You saw this bacteria. It created this passion in you, and you brought it with you all the way west to a place where there were no ticks that were harboring that bacteria, at least at that time. And we'll talk more about that later. But to talk about how that spurred this passion in you, and how you brought that back with you all the way to the western part of the U.S.
Well, you know, so I was influenced by the colleagues in my department when I moved to Salt Lake City, and in my department and around me. And so in the Department of Pathology, there were a couple of other faculty who were outstanding mouse immunologists. And because Borrelia burgdorferi, the natural reservoir is the wild mouse, and Steve Barthold had just discovered that there were inbred strains, there were laboratory mice that could be used to study the diseases caused by, the symptoms caused by Borrelia infection, so the arthritis and the carditis. I thought it would be a really good opportunity to study Borrelia infection in the mouse. And so these colleagues were very helpful in getting me started working on a Borrelia infection in the mouse. And I initially thought, well, we need to look at the immune response, and we need to study how mice respond to infection with Borrelia burgdorferi.
Okay, so let's pause there for a second. We want to define some terms here, because this is important for our community. So first, please define what a pathologist is and what an immunologist is.
Okay, a pathologist is generally a physician or a DVM, a veterinary person who studies the tissue responses to an infection or any kind of disease. It might so studies the abnormalities that can occur following a disease. And an immunologist is somebody, a scientist or a physician, who studies the immune response. And this can be an immune response to a bacteria or a virus. It could be an immune response to cell tissue, an autoimmune response. And so immunologists have a lot of constantly developing new tools to understand how the immune system, our immune system, responds to any kind of challenge, be it an infection or an autoimmune challenge, and trying to understand in more molecular details how things go correctly, how we appropriately fight an infection, or in some cases how things are sidetracked and we have an inappropriate response that can lead to a pathological outcome, and that's where the pathologist would come in.
Okay, and give us one more definition. What is a microbiologist?
A microbiologist is someone who studies microbes, microorganisms. And so my PhD was in microbiology, and I have studied bacterial infections. And I study, and a microbiologist can study how bacteria function, how they divide, how they make toxins, how they are select for certain growth environments. My studies and background are more in the interactions of microorganisms with their hosts. So host pathogen interactions. And so I've always studied bacteria that can infect people.
Okay, so when we're talking about hosts, so for example, if I were bitten by a tick and the bacteria that you study was spit into me, I would be the host, and the bacteria would be the organism that you would be studying as a microbiologist, meaning my response to the bacteria, correct?
Right.
Okay. And an immunologist would be studying my immune response to the bacteria that was spit into my system, right? So that, again, as the host, you would be studying my immune response.
That's correct.
And then, of course, a pathologist would be looking at the impact that the bacteria was having when it was spit into me, the host, and my immune response, or the impact that my immune response was having to the tissue. Give me that connection between the pathologist and the immunologist and the microbiologist.
So the pathologist then would look at the abnormality. What's abnormal? So for my research, we're very dependent on a veterinary pathologist, Jim Zachary, who actually looks and evaluates heart tissue or joint tissue from infected mice to say, well, and he's got a way to rank and say, he looks not only at the cells that are changed, he looks for changes that would be abnormalities caused by the bacterial infection. And they could be caused by products the bacteria make that are detrimental to the host, in our case, the joint tissue. He would look for immunological cells that infiltrate due to the bacterial infection interacting and causing changes in the host tissue. So the pathologist sort of looks at the end product and says, these are the things that are wrong and that are going wrong, and these are what's causing, these are the characteristics of this arthritic joint. Whereas, and a lot of those are likely to be mediated by the host's immunological response to the bacteria.
So one of the things that Dr. McDonald, when we interviewed him, he described a pathologist as sort of a detective, right? He called himself the doctor's doctor, where they're looking at the impact that the disease process has had on the host. And then you can work backwards to determine how that end product had occurred. Is that a proper description?
Yeah, I think that would be. And yes.
Okay, so now let's talk about the mice, right? I mean, you arrived at the University of Utah, and you now have this unique experience where mice are now available to you, and they're available to you in your research in a unique fashion. So build that out for us, because we understand that mice are a very important part of the team that ultimately helps the Lyme bacteria become sort of supercharged, and when it gets spit into us and the impact that it has. So talk to us about mice and their role in the disease process.
Right. So in nature, the wild mouse is really critical because it's a major reservoir for the bacteria Borrelia Burgdorferi, and Borrelia infection of mice is really interesting. The mice can harbor the Borrelia for many months, even though, whereas many other bacteria would be cleared by the mouse's immune response, Borrelia are very good at evading the host defenses, the mouse's immune response for clearance. And that's critical because an infected mouse is essential for the tick during their feeding stages, the larval and nymphal stages, to feed on a mouse that actually infected with Borrelia Burgdorferi, and for order for the tick to become and pick up the spirochete, the bacteria, and become infected and competent to transfer to another host at the next feeding time. And so
let's stay with this mouse in the wild for a moment, then we're going to get to the mouse in the research lab. So it seems like the bacteria and the mouse immune system have developed some kind of a symbiotic relationship, because the bacteria is able to evade the immune system of the mouse, yet it doesn't make the mouse sick. Talk about that, how the bacteria and the mouse immune system seems to have developed a symbiotic relationship where the bacteria can stay harbored in the mouse without making the mouse sick.
Right. So, there's some really interesting new research in this area. It's something we really haven't understood. We just know that in wild mice, they become infected with Borrelia, but they don't display arthritis or carditis symptoms. Alan Barber, who of course was one of the first people to research and discover Borrelia burgdorferi, was working with Willy Burgdorfer when it was first cultured. He has recently made discoveries studying the wild mouse and found that if you look at the wild mouse's response to Borrelia burgdorferi, it's really it's very it's muted. It's not a robust inflammatory response. There seems to be truly we don't know if symbiosis is a proper word, because we don't know what Borrelia does for the mouse, if it does anything positive, but the this mouse's response to Borrelia is different than the laboratory mouse's response, different from human response to Borrelia. And it seems to be, and it's not just to Borrelia, it's to other kinds of pathogenic bacteria as well. It's just a less it's just a low inflammation response, sort of, and it seems to cause very little change in the mouse.
Dr. Weis,
much less inflammation.
Dr. Weis, I'm just curious about, we talked about the term pathologist with Rich, right? Where you study the tissue response to a pathogen. And we talked about how in humans, of course, we know our tissues respond to Lyme disease, and we have inflammation and we have all kinds of responses to it, and we can get very sick. And you just described that in a mouse model, in the wild, a mouse generally is not impacted by the Lyme bacteria, but in the research model, it is, right? So
Yes.
from working with a pathologist, what is the viewpoint from a pathologist why humans have such a response to the bacteria and it impacts their tissues negatively, and in the wild it doesn't, but then in the laboratory environment mice do have a response to it? It just seems so odd to me that there's three scenarios with kind of three different reactions to the bacteria, right?
So, initially Stephen Barthel did a lot of work when he was at Yale, and he was a very distinguished veterinary pathologist and Ph.D. DVM, and had developed a model for mouse hepatitis and had devoted his career to understanding pathology and mouse infections. And so he was in a wonderful position to look at the response of wild mice to Borrelia burgdorferi, and he saw we're not seeing anything that represents and mimics what we see in people. But he knew that laboratory mice could be could provide opportunities, because laboratory mice they're sort of, it's almost like thinking about different breeds of dogs. Even though laboratory mice all look about the same size, they're different, and they have different versions of genes that are shared by all the mice, but different, we call them alleles, different versions of these genes, and some combinations that are found in different laboratory mice make them much more susceptible to very different types of infectious challenges, cancers, eye abnormalities. There's just this spectrum of and at that time, dozens and dozens of different laboratory mice that are slightly different from each other, but generation and generation, a particular mouse strain, we use one called C3H, is reproducibly shares certain traits and inherits certain traits. And so Dr. Barthel looked at 16 different laboratory mice and infected them all with Borrelia burgdorferi, and he could find a spectrum of disease. And he found some laboratory mice that developed very severe disease. This is the C3H mice. He found other laboratory mice that were reproducibly had very mild disease, the Black 6 mice, and they looked like the black 6 mice looked much more like the wild mice. And so he was able to say, well, when we can kind of isolate these versions, various versions, and keep them constant in different laboratory mouse strains, we can actually look and see, when we infect some of these mice, pathologies that look like what we see in humans, in patients.
Okay, so just real quick, I want to just build that out a little bit more. So we know a big part of what you're studying, Dr. Weis, is how genetic elements can impact illness and the mouse model with Lyme disease, right? And you just use some high-level words I want to break down further, because wild mice, again, not so much, they don't really get sick. Humans, they can get, some humans don't get sick at all, right? They're asymptomatic.
That's right.
Some humans get extremely sick and become chronically ill. We understand there's co-infections, other things, other genetic pieces to it, but I think they're all related, right? So when you said that in the laboratory environment, the C3H mice react very severely to Lyme disease. However, I think you called it the black B mice, are reacting very mildly to Lyme disease. Are those genetic expressions? What does that mean, C3H mice versus black B mice? And what is the role that genetics play in having these hosts, the mice in your research, respond differently to Lyme disease?
So, so as you mentioned, some people have very mild symptoms and others very, very severe symptoms. And a great example of that is forestry workers. There's a lot of incidents, several publications showing, old and even more recent, that certain forestry workers will have evidence, serological evidence, antibodies in their blood indicating they've been infected with Borrelia burgdorferi, but no evidence, no remembrance of any kind of disease. So that's an example of somebody who was infected, but the infection was so mild that it was not followed up, or they wasn't recognized. Whereas other individuals get severely ill, acutely, and then chronically ill. So the identification of mouse strains that display a sort of a spectrum of disease severity was critical for saying that perhaps this could be a model for these different responses that are dependent upon the host, in the inherent genetic composition of the host, and why so it provided an opportunity to say, maybe we could compare the C3H, and it's actually black 6, I'm sorry, the B6 or black 6 mouse with the C3H mouse, in order to try to identify what are differences in the genomic inherent genes, different versions of these genes that are passed down and inherited by these two strains of mice. And I also really want to acknowledge that we know that there are also different isolates of Borrelia burgdorferi. A lot of great studies have been done to say some of the Borrelia burgdorferi only give a skin lesion when they infect humans. Others give this disseminated disease and can cross the blood-brain barrier and cause arthritis and carditis. And so we know that there's clearly the bacteria themselves have genetic differences that contribute to disease severity. So but the nice thing within laboratory model is that you can use a single isolate of Borrelia burgdorferi, and you know that all of your mice receive the same number of bacteria, have the same isolate, the same clone of Borrelia, and yet they have different responses, different severity of disease when they're infected with that.
Okay, so just to recap that, so we know that Lyme is very complex. We know that there are various species of the Lyme bacteria that impact the host, meaning humans or mice, differently. Some can cause just arthritis, some can be disseminated, cause heart issues, brain issues, neurological issues, etc. But what's so, I think, beneficial about your research is you are isolating a specific strain of Lyme disease to test on all of your mice, and you're getting different responses based on the genetic sequence of these mice, which are the black 6, the C3H, etc. And depending on what their genetic expressions are, they are responding differently to the same strain of the bacteria. Is that correct?
That's absolutely correct.
Okay, so now we know that's just something we're born with. Our genetic sequence, our genes are what we're born with, but we also know you study epigenetics as well. So we have the genetic, just sort of you're born with what you're born with kind of component, but then epigenetics are how your genetics are sort of turned on and off or affected by external environmental factors. So what have you learned, Dr. Weis, about epigenetics when it comes to Lyme disease, studying the mouse model?
I guess we haven't done a lot. We have looked at microRNAs, but they are also genetically inherited. So, but their expression is regulated in response to inflammatory stimulus. I would say that my lab has really not, we've done some epigenetic studies, but we really have not published and brought to completion any study on epigenetics. So I think that's not the contribution that my lab has made. We've really focused on the genetic, and some of these genetic differences may be mediated through epigenetic alterations, certainly, and through differences in the ability and the magnitude of response. But our focus has been, and what so far has been, on actually gene sequences that are distinct between the C3H and black 6 mice. I'm sure epigenetics, I mean, in fact, I know epigenetics are important because we've seen some similarities reported in patients for some of the same genes, but we haven't gone there.
All right, so let's talk about genetics versus epigenetics, because I'm fascinated by this topic, right? I mean, human beings are adaptive beings, right? I mean, it's one of the reasons why we are the dominant species in the world, because we're so adaptive, right? And when the Human Genome Project had taken place, I remember, again, as old as I am, I remember when the project started. I remember all the excitement around it, and I remember we came up with, we sequenced our genes. And I remember at the time when it was first reported, we had this sequenced set of genes, and we had what I think they were calling like junk genes in the beginning, right? Where they didn't know what it was. And as it turns out, I think most of the researchers are now coming to the conclusion is that we certainly have encoded genes, and actually we have fewer encoded genes than I think an onion does, which is interesting. And then we have these genes that are not encoded, which are the genes that allow us to become adaptive to our environment and to our world, right? So the epigenetics is that set of what used to be called junk genes, or the genes that didn't have a purpose, which allow us to become more adaptive and allow us to have a response that permits us to survive in diverse environments. Is that accurate, or would you give us another description of what epigenetics is?
So the epigenetics are changes in the way the genes are expressed, and they can be regulated by biochemical changes to the DNA in a person's particular cell. For example, there are modifications of DNA which influence the magnitude at which the gene that is encoded by that piece of the DNA is expressed. So we go the gene in the DNA, and then you go to messenger RNA, and then you go to protein. And there are many things, like you're talking about the previous junk DNA, things like microRNAs that are encoded in the genome, and then the RNA comes back and it sits down on a transcript, and it influences the microRNA influences the magnitude to which a gene can be expressed. So it's encoded someplace else, and one microRNA can influence the expression level of dozens of different genes in an inflammatory process. And my lab did study a microRNA in that regard. Certainly.
Okay, so let's talk about that as a practical matter, Professor. So epigenetics can be a gene being turned on or off, correct?
Yes.
It could be epigenetics could be a gene being enhanced, meaning the expression is greater as opposed to not as aggressively expressed, right?
Yes, absolutely.
And it could be turning on a gene that had no function at all, now having a function in the body of the host.
It could influence. It also could cause re-expression of something that's supposed to be expressed only, say, during embryonic development.
Okay.
And that would be something I think would be that people studying cancer are very interested in, turning on of embryonic developmental genes at a later time or in inappropriate tissue. So altering the regulation. Yeah. And
then it will have an impact on the way the different genes interact with one another?
It could.
Okay.
Yeah.
So give us some insight again from on a practical level, not in the scientific level, but in a practical level, what impact epigenetics is playing in the way that our bodies as hosts are responding to the Lyme bacteria.
So in the case of microRNAs, there are, let's see. So when we are infected with Borrelia burgdorferi, and our inflammatory cells, white blood cells, respond by making large amounts of particular molecules designed to fight the infection. These are very important and helpful at starting inflammation, which can result in clearing the bacteria, but they have to be turned down. So they have to be, they can be expressed very robustly, but they can't go on forever expressed robustly. They need to be regulated. And so simultaneously with turning on inflammation in a good way, there are also other genes that encode the molecules called microRNAs that are turned on. And the microRNAs actually go back and they kind of act like a rheostat. They don't shut off, but they temper down the level. So they regulate and they say, okay, we don't want to have overabundant inflammation because that can be very harmful, especially if it's localized in a particular tissue like. But we can suppress and kind of temper it down and turn it down. And so these are things that are turned on simultaneously with inflammation, but they act as sort of a feedback just to rheostat, to reduce the level of inflammation to an appropriate level that's not harmful to the host. So you want a very robust initial response, but you want to kind of control it.
Okay, so let me walk back before Matt is getting really excited and he wants to jump in and geek out with you on some of these issues, but I want to walk it back to the probabilities, or I guess maybe permutations. So one of the things that we've seen in our community is, and I guess we could even use me and Matt as examples, is we have a great deal of diversity within the human population, just like you have the diversity within the mouse population, right? And in the human population, we have a whole bunch of people, as you had pointed out with the forestry studies, where they have come in contact with ticks on a regular basis, have been bitten by ticks on a regular basis, and they have no response. And I think I may be one of those examples. I've been bitten by ticks in my entire life. I grew up on Long Island, and tick removal, and we had a tick kit sitting on the shelf that we walked in the room when we walked into our house in our mudroom, because we were bitten by ticks so often, and our companion animals were bitten by ticks so often that we had to remove ticks all the time, right? Yet thankfully, I'm knocking on wood, I've never been sick. Whereas now we have Matt, by contrast, who also grew up on Long Island, and he became chronically ill. So that could, of course, happen in one of two ways. One way it could be is, as a host, I may have a different set of genes that are allowing me to respond differently, or when I'm being bitten by a tick, the strain of the bacteria or the combination of germs that are being spit into me may be different than the combination of germs that are spit into Matt, right? So you're seeing the same thing with the mice. So one of the questions that was sort of jumping out at me is, why do you believe the mice have the different genetic expressions? Is it because they're from different regions of the world, or because they've come in contact with this pathogen and they have a built-in sort of immunological software that's allowing them to respond? I mean, what are your sense of why you're seeing the diversity in the mouse population, and does that apply to the human population?
So I think in nature, presumably the wild mice that would get very sick when infected with Borrelia burgdorferi, they would be selected against because they wouldn't. And so the mice that we see are ones that have a moderated response that allows them to not become disabled upon infection. that there's just the selective pressure would be too great.
Right.
In the
But you're seeing-
laboratory mice,
Right. In laboratory mice, so you're seeing a spectrum, right? Is that because they've been bred to have a spectrum, or because they were from a diverse population of mice that were captured in the wild and you saw these different presentations?
most of the mice derived decades ago. They are initially were derived from these fancy mice that were kept by royalty, and they were sort of amateur hobbyists, just sort of characterized. Oh, look, these mice are all white. These mice are black. These mice are this brown agouti color, and they were initially bred as fancy mice, but through inbreeding, they were selected to have different traits that were also followed up. So there are dozens and dozens of strains, but they are relatively similar to each other, but they have different traits. And there's no selective pressure for laboratory mice that are now bred and housed under pathogen-free conditions with all the food and water and clean bedding that they need. So there's no selective pressure that would cause loss of genes that were slightly defective or made them slightly less competitive, so they could be maintained. And now, they've been for decades kept at Jackson Labs and bred at Jackson Labs in Bar Harbor, Maine, and so they've been genetically identified as genetically pure and distinct from each other by sophisticated types of techniques, and they are constantly being monitored for the appearance of mutations that would alter them. But the selective pressure is off because they live in a controlled environment where they wouldn't be selected against. So that's why so they were initially derived for traits that were unrelated to bacterial infections, but they coincidentally, along with the traits that they were being selected for, we found the other kinds of differences. So some mouse strains are very prone to cancer, as I mentioned, and other types of defects. So the C3H and the black 6 mice, they're pure and separate from each other and inherited, but the ability or this trait of the C3H mice to have severe disease when infected with Borrelia burgdorferi, it's not selected against, so it can be maintained. And so our hypothesis was that the spectrum of disease that we can see and compare, if we looked at the two most extreme examples that Dr. Barthold identified, the C3H and the black 6, perhaps we could compare the response of these two mice to a single Borrelia, isotype of Borrelia or isolate of Borrelia, and identify what are the differences that allow the C3H mouse to develop severe disease and allow the black 6 mice to be resistant to that disease.
So Dr. Weis, when you say selective pressure, does that mean that the C3H mice in the lab, it's going to survive and there's a benefit and a use, but in the wild, it would become very sick and it wouldn't reproduce and it would die off because it has such a poor response to these pathogens? Is that what you mean by selective pressure? I'm not sure what you mean by that term.
Yeah, that is exactly what I mean by selective pressure, that there's nothing selecting against a defective response to a particular pathogen or a skewed response or an abnormal response in the lab.
And let's jump back to when you're talking with Rich about certain genetic expressions or genetic activations during the embryonic phase, or when you're being developed in your mother's womb, versus genetic expressions when you are an adult or a young person. So that made me think about certain genes possibly related to stem cells and things like that. So can you expand upon the role of genes during the embryonic phase and the work being done now and the research being done now to see how we can activate those genes to further enhance, adults, as you were saying? So it sounds very interesting in using that technique or model or thought process to help the health of humans today, and maybe in regards to Lyme disease as well.
Boy, I'm getting kind of out of my area of expertise. I'm sorry. I'm sorry. I'm not a stem cell biologist at all. And there are people who, we have a stem cell core at the University of Utah, people who really think about these things and how to use stem cells to correct, defects, autoimmune defects, for example, and things. But it's really out of my expertise. I mean, and so I'm sorry, I just don't want to give wrong information.
No, I certainly appreciate that. I mean, that you gave us more than enough already in that regard. So, again, following back with something you talked about with Rich, you kept talking about the microRNA, and you said when somebody has an inflammatory response, that's a good thing because you banged your elbow, right, and you're having an inflammatory response, but you need something to sort of downregulate or calm down that inflammatory response
right.
as time goes on. And I believe you said that was the microRNA that assists in that process. But I know a lot of our listeners are going to be thinking, first of all, what's RNA and how is it related to DNA is the first question I have.
So our genes are encoded in our DNA, and the DNA is a copy of it's the genetic blueprint to make all the proteins. But in between the DNA sequence in the genome, there's a molecule called RNA, and RNA is transcribed for all of these genes, and the RNA then goes to the ribosome, and the ribosome translates the RNA into protein. So DNA to RNA to protein is the fundamental process by which we make proteins, which in our cells. MicroRNAs are kinds of RNAs made that don't encode proteins, but they do encode these regulatory sequences that regulate the expression of proteins through causing degradation of the RNA. So you go DNA, RNA, protein. The microRNAs come back and bind and find particular RNA and reduce the expression, the translation of the RNA into protein at the ribosome. So again, they're specific. They don't for particular RNAs. And the ones we've studied in my lab, Rob Lockhead was a graduate student who took a lot of initiative and collaborated with Ryan O'Connell, who had recently joined our department. And he studied an RNA called microRNA 146A. And what microRNA 146A is an anti-inflammatory microRNA. So during an inflammatory response, you get a lot of inflammatory genes activated that go DNA, the gene becomes activated, you get transcription of the RNA that goes to and is translated into protein. The microRNA that's also produced at the same time as the RNA for the inflammatory genes comes back, the microRNA finds and identifies the transcripts and actually specifically binds to the messenger RNA and causes it to be degraded, thereby tamping down. So the microRNAs are stimulated, synthesized, and they at the same time as the RNA for inflammatory molecules, but they turn it down. They dampen the response.
So they're complementary processes. So it sounds like the first, I bang my elbow and I have an inflammatory response. The DNA tells the RNA to create a protein that goes and creates the inflammation. And I'm oversimplifying this just so I can understand it, right? But in parallel, a complementary process is the DNA then turns on this microRNA, which says, hey, we need to go down regulate this protein to decrease the inflammation so it doesn't become a problem, and we only get the beneficial side of the inflammation because of the elbow bang. Is that at a high level what you're saying? Or
yeah, I think so. Yeah.
Okay. So, you also talked about earlier the autoimmune component. You mentioned that a couple of times. And I know from doing some research and studying about the work that you've done, you are doing research to look at chronic conditions that are potentially involving autoimmune pathways with studying genes, right? So can you just give us more information about the work you're doing and how it pertains to studying autoimmune complexes? Because so many people listening to this podcast are dealing with autoimmune diseases or autoimmune symptoms because of being diagnosed with late-stage Lyme disease or another tick-borne illness.
So so one of the things that we identified in the mouse model, in the C3H mouse, was the overproduction of type 1 interferon. And we have shown that in C3H mice, that if we, that at 1 week of infection with Borrelia Burgdorferi, in the ankle joints, there's a huge upregulation of type 1 interferon and the genes that it regulates. And then that goes, if we actually treat a mouse with an antibody that blocks type 1 interferon, we can reduce the severity of the arthritis. Type 1 interferon is really interesting because it is intimately connected with lupus, systemic lupus erythematosus in patients and in animal models. And so in lupus, we have a lot of autoantibodies that drive the development of disease. Now, I should say in the C3H mice, we think that the type 1 interferon is driving inflammation, but you don't have to have any autoimmune, any T cells or B cells in a C3H mouse in order to get severe Lyme arthritis. That's an experiment that has been done by several individuals, including ourselves, and we've shown that the arthritis you see in C3H mice is not an autoimmune driven. That doesn't mean that there aren't autoimmune driven arthritis and that there's not involvement of B cells and T cells in other types of models and in patients, but we can just separate the impact of the type 1 interferon in C3H mice and show that it is driving an inflammatory response that's not an autoimmune response.
So just to clarify, B cells and T cells are immune cells. So when you're saying that in the C3H mice, which are the mice that have a severe reaction to Lyme disease in the lab model, they do have arthritis, severe arthritis, but there are no activation of immune cells like B cells or T cells, which means there's no autoimmune portion to the inflammation. They're just getting inflammation from another way, I think is what you're saying. Is that correct?
I want to clarify that.
Okay.
Certainly there's activation of B cells and T cells, especially B cells in a C3H mouse. But we can have a special breed of a C3H mouse that lacks all T cells and B cells, and they still get arthritis.
Oh, so you're able to isolate the fact that it is not an autoimmune response by really specializing and refining your sample study of the mouse.
That's correct. So definitely you get a huge in a C3H mouse that's not been modified, that does have an active B cell and T cell component. You get a lot of antibodies made, but the antibodies themselves are not driving the disease. So that's a very confusing. So just because you have this heightened production of antibody doesn't mean that's responsible. You have to separate, is that causing it or is that a consequence of the bacteria and its potent ability to stimulate inflammation and stimulate B cell activation? So that, yes, that's the clarification. It's tricky to think about it.
Well, that's the really cool part about your research though, right? Is that you're able to do this modification to see really what's causing what, because you would think, well, there's a ton of immune response, so maybe it's autoimmune. Well, it's really not, because you were able to modify it. How are you modifying the C3H mice? Are you suppressing the immune system through some sort of drug and then proving that the arthritis is still present? Like, what's the process to do that study?
Okay, so this is something that, hundreds of researchers have worked on, and there are C3H mice that have been developed that have a mutation that prevents the development of B cells and T cells. So, and there's
a genetic mutation.
A genetic mutation in the genome. You may have heard of them called SCID, severe combined immunodeficiency. They're mice with a severe combined immunodeficiency. Just like, you've heard of ages, the boy in the bubble that has a severe combined immunodeficiency. And nowadays, children born with that would get a bone marrow transplant and hopefully do very well. But those genes were studied that were identified first in patients, but in mice and there's several different gene mutations that can cause a mouse to have a severe combined immunodeficiency. So it's combined because they lack both the B cell response that makes antibodies and the T cell response that helps antibody production and is important for antiviral responses. So we could do infections in the laboratory mouse where the mice can be kept healthy because they live in a pathogen-free environment. You can do experiments in a severe combined immunodeficiency mouse, different flavors, infect with Borrelia Burgdorferi, and you can see, well, those mice have, they still can develop arthritis. So the arthritis does not require an autoimmune response between the host recognizing host B cells causing antibodies or T cells that recognize itself. It's actually driven by the bacteria invading the joint tissue and causing the arthritis locally in the tissue.
So that's what I was going to ask. So it's not the inflammation causing the arthritis, it's the actual Lyme bacteria, the spirochetes, that are infecting the joints and causing inflammation and causing the arthritis, is what you're saying.
That's right. So we know it's the inflammation that the bacteria cause in the joints. And this is the beauty, though, of being able to compare a C3H mouse with a black 6 mouse. So the C3H mice and the black 6 mice, both with the same type of mutation, they have similar numbers of Borrelia in their joint tissue. But the C3H mice gets really severe arthritis and the black 6 mouse does not. So it's the genetics. That's what says it's the host genetics that determines the severity, the magnitude of the inflammatory response to the bacteria that invade the joint tissue. It's not just the bacteria's ability to invade the joint tissue, it's also the regulation of that, the level of the inflammatory response.
So have we in your research figured out why these black 6 mice are not getting as sick, meaning the arthritis is not as bad, but yet it's the same concentration of bacteria, and that same bacteria is in the joints, eating away the joints, infecting the joints, but yet these C3H mice are getting horribly sick with arthritis, and the black 6 mice are not, right? So what are the genes doing to actually help these black 6 mice, right? Because the inflammation is still really there in the C3H mice, but not so much in the black 6 mice, right?
So we have studied this for 30 years. We have had a project, it's called Forward Genetics, where we made black 6 mice and C3H mice. And we did complex crosses between these mice and their offspring to identify in an unbiased manner, so without preconception, to say, are there genetic regions of the C3H mouse when inherited that are associated with more severe arthritis when we infect with Borrelia Burgdorferi? And in doing that, in collaboration with an immunogeneticist, Corey Tusher, through the years, my lab has followed and identified several different genes that are, if the allele for that gene comes from C3H, the offspring develop more severe arthritis. So we were able to isolate different regions of the mouse genome that regulate arthritis severity. One of the most exciting and most recent discoveries, which has been worked on through the years by several different students and postdocs, is a gene that regulates type 1 interferon. And the type 1 interferon is an antiviral molecule that can also cause detrimental pathology if it's highly expressed in a particular tissue. And in C3H mice, type 1 interferon is highly expressed when Borrelia invade the joint. And we've identified a gene that regulates type 1 interferon that had not been associated with interferon-induced diseases previously. So we're really excited about that because it offers a target for treatment of severe Lyme disease, and type 1 interferon elevation has been identified by several investigators who are studying Lyme disease in patients. Neuroborreliosis, skin, blood cells have all been identified in patients of having elevation in type 1 interferon. So we're very excited about that.
I definitely want to ask you more about the interferon piece, but I guess where I'm a little confused is we understand that mice that have this C3H gene, or I think this genetic component, they're getting more sick. The black 6 mice are not. We know there's that correlation, but we understand exactly why the mouse is getting sicker in the C3H model versus the black 6. Other than saying, well, if they're black 6 mice, they're not going to get as sick. If they're C3H mice, they will get as sick. Do we understand exactly what's happening that's causing that process of sickness to occur in the mouse?
That's what we're continuing to study. So we know type 1 interferon is really important in the C3H mice. We know if we have a genetic a mutant mouse that doesn't make type 1 interferon, they don't get as sick. We know if we can give them an antibody, a blocking antibody, and that we can reduce to type 1 interferon, we can reduce the severity of disease. We also know now that if we're looking, what is type 1 interferon doing to induce the development of arthritis? This is sort of a novel concept because we know it's not dependent upon the same things that happen in lupus where you get a number of autoantibodies. So what's type 1 interferon doing? We have one hint because we can do genetic studies and looking for expression patterns of of cells actually in the joint of a C3H mouse that's developing arthritis. And we know that there's a gene called myostatin, which is a muscle regulatory gene that we would never have expected to be involved in Lyme arthritis. And in fact, a previous graduate student, Jackie Paquette, identified myostatin as being uniquely upregulated in response to Borrelia. Myostatin is, so now we were able to collaborate and use an inhibitor of myostatin and cause a reduction of the severity of Lyme arthritis. So we are developing kind of, by teasing this apart, we're going A to B to C to D. And that's what, the goal is to try to find elements that we could target selectively in Lyme arthritis development, new targets that could be used in patients that would reduce the severity of arthritis. And these are things that are not targeting the bacteria. So they're additional, they could be used in addition to antibiotics for resolution of disease, but looking at the inflammatory processes that have been just so robustly and possibly inappropriately activated in certain individuals to try to reduce the severity of disease.
So what I like so much about this is we always talk about how Lyme, especially chronic Lyme, is so bio-individual because we never know what's going on in each of our bodies. But this can be a really nice supplemental aid to any kind of treatment that a Lyme patient chooses to help reduce inflammation, to help regulate these musculoskeletal genetic expressions, to give the body a fighting chance to decrease inflammation, support your specific genetic makeup while treating, which gives you a significantly higher chance of recovery when dealing with chronic Lyme, is what you're saying, I believe, right? I mean, this is really great personalized treatment to aid in the other treatment that's out there today. But just for clarification now, so the type 1 interferon, you said, earlier you said it's an overproduction of type 1 interferon, which drives inflammation, which is different than the lupus part, because lupus is driven by immune cells, the B cells and the T cells, right. and the interferon really drives inflammation, but it's not really coming from the immune system. So what is type 1 interferon? Is it a hormone? What exactly is it, and how is it causing inflammation compared to the lupus side of things with Lyme disease, which is more an overreaction of immune cells causing inflammation?
So type 1 interferon it's called a cytokine. It was it's called interferon because it interferes with virus replication, and it was first identified for its antiviral effects. It's a very potent antiviral molecule and it's but it's also very potent in promoting inflammation. So interferon acts antivirally by it is a hormone. It's a cytokine, but it has hormonal activities. So interferon is made in one cell very shortly after viral infection. It's secreted by that cell, and it can come feedback on itself. So that would be autocrine. It can go to an adjacent cell and through an interferon receptor and cause the antiviral state in an adjacent cell. And that's probably its most important activity. Would be called, so that's a hormonal activity to cause nearby cells to turn on an antiviral state. And so the antiviral state shuts down the ability of the virus to be replicated by the adjacent cell, because a virus comes in and takes over the cell's nucleic acid, DNA, RNA, primarily RNA production and protein production. So you turn on the antiviral state to shut down the production of virus. So interferons are very, very important in the initial control of any viral infection. But interferons at high levels, but they're also tightly controlled. They boost on and they come back on. If interferons are present at high levels, they can make people really, really sick. So we know, for example, hepatitis or multiple sclerosis, people can be treated with interferons, and it helps control a viral hepatitis infection or the immune phenomena of multiple sclerosis, but it makes people very sick, and some of the consequences include arthritis in those patients. That's why the new Gilead treatments for hepatitis that allow that now no longer require the use of interferon for patients with hepatitis have been game changers. It's just tremendously improve the quality of life of individuals who have been treated for hepatitis, because interferons can be so detrimental. The consequences, side effects can be so detrimental.
So it seems like Lyme disease in general creates so many things to be off balance in your body. We've learned from other professionals that are studying hormones in Lyme patients that in many Lyme patients, cortisol production gets stuck on overdrive, and your cortisol receptors aren't responding well, and your body keeps producing more and more cortisol, and it has a really negative impact on your hormones. You have adrenal fatigue, you have burnout, it has a whole component there. And what we're hearing from you, Dr. Weis, is that interferon is a necessary component of the human body as a process, and it's good when it's turned on to address some sort of, as an antiviral to address a virus, but then it needs to shut off because it needs to be regulated in the quantity and the duration. But in Lyme patients, it's almost getting stuck on overdrive, where it's just always on in high concentrations, and that's contributing to inflammation, causing pain and discomfort in Lyme patients. Is that correct?
Well, in patients, we know that there's evidence that it's on long term, or that can be identified. In the mice, it goes on at a very hyperdrive level, and then it turns back. It is turned back down. So it's on at 1 week of infection, comes back down, but the arthritis doesn't develop until 4 weeks later. So we think it's the consequence of that really strong boost by activating things like myostatin that lead to the development of the arthritis. But once the arthritis is full blown, it's hard. It takes a long time for that arthritis to resolve, even if you were to treat the mouse with antibiotics. That inflammation is very stubborn to clear.
So did you say, Dr. Weis, that in humans we know that it gets stuck on, but in mice it does not? But even in mice, that when it spikes pretty aggressively and it comes back down, weeks later it causes severe arthritis. But in humans, if it gets stuck on, it can be even worse. Is that what I heard? I just want to make sure I'm hearing you correctly with that.
Well, it hasn't been studied. Type 1 interferon hasn't been studied. It's a marker that has been identified and reported to be elevated in patients with neuroborreliosis, to be elevated in blood from patients with early Lyme disease, for example, with skin lesions, things. So it's been reported, but its regulation has not. And so I don't want to overstate. We think that it is on. We know that there are reports of type 1 interferon being induced in Lyme disease in patients, but its regulation has not been studied in those patients, and it needs to be studied.
Meaning we know it's definitely been on and at a high level. We don't know if it's staying on or for how long it's staying on, as we're saying in the human model, right? Okay.
That's right.
Gotcha. But we don't even at a spike, when it comes back down, it still causes severe illness in the mice model with arthritis. So I guess where my head is coming from with this is, in the Lyme, we have some Lyme patients we interview who say, I have no autoimmune issues. I've had all my blood work done, but I still have severe pain, chronic pain, and arthritis. And that could be potentially related to an overproduction of type 1 interferon, but then the lupus piece of it, the autoimmune piece of it, is separate and distinct, where you're having an immune response. And then there are other patients who have chronic pain due to Lyme, which it could be coming from the autoimmune response with the T cells and the B cells. Is that what you're seeing in your research, Dr. Weis, that there could be two different triggers for inflammation and pain with Lyme patients that I just described?
Yes, that we actually, and we have a model for T cell involvement and it's a different model.
That's okay.
It's a model on the black 6 mouse. It's a genetic model. It's called the IL-10 deficient black 6 mouse, and IL-10 is it's a cytokine that's an anti-inflammatory cytokine, and if you lack that cytokine, the mice have a much higher, they have a high B cell response, but they have a very active T cell response that's making a molecule called interferon gamma. So it's not type 1 interferon, it's a different kind of interferon. And it seems to be really important in the T cell mediated disease that's seen in some patients. So we know that some of the chronic Lyme patients, patients with post-treatment Lyme disease, that in their joint tissue, they have high levels of interferon gamma, not type 1 interferon, but interferon gamma, and a number of other cytokines. And they have a lot of T cells and inflammatory infiltrate that is sustained after the treatment with antibiotics. And in our hands, we have the IL-10 deficient mouse, and we have looked and we have similar sustained arthritis, hardly non-detectable Borrelia still in joints, even though these mice haven't been treated with antibiotics. In the absence of IL-10, the inflammatory response is so strong, it really it clears the Borrelia very efficiently. And they produce autoreactive, in humans, autoreactive T cells and antibodies. In mice, we have sustained bystander activation. All kinds of T cells are activated in the joint tissue and in the mice and in the lymph nodes to the joint tissue of the mice. And we think it's promoting an environment that in the right conditions, an autoreactive autoimmune event could occur. At this point, what we're tracing is just non-specific activation of all T cells in the region, and that's producing cytokines of inflammatory molecules that cause recruitment of white blood cells to the joint tissue, which are really that make this pro-inflammatory environment, and that's really what we're seeing is driving the chronic arthritis in our model of T cell driven Lyme disease. So we have two different models. So this is one that's definitely T cell dependent. It causes a non-classical activation of T cells. It's not antigen specific T cells, it's all the T cells that get activated. And we think it's a direct effect of the ability of the Borrelia burgdorferi to get into the lymph nodes, activate cells in the lymph nodes, and also the Borrelia to get into the joint and then recruit T cells to the joint tissue, and then cause this very active inflammatory environment in the joint.
So, Dr. Weis, why is the inflammatory environment an environment that benefits the bacteria?
I'm not sure if it's an environment that benefits the bacteria. The bacteria are present at very, very low numbers to non-detectable in the joint. It's just a lack of control, a dysregulation, and so I don't know that there's an advantage to the bacteria. It's just we're using the IL-10 knockout mouse as an example of an environment that's lost regulation, and we don't think that patients totally lack IL-10, but they have a dysregulation. The patients who after treatment have sustained symptoms, something is out of regulation, and we'd like to figure out ways to reset the inflammatory response in the patient.
So why does the inflammatory response have an impact on our ability to clear the germs? And if it does, then doesn't that benefit the germ?
So in this case, in the patients, they've been treated with antibiotics. So the bacteria should have been removed. So there's something preventing the inflammatory response from returning to baseline, returning to normal. So is it due to the persistence of fragments of the bacteria, components that are not being appropriately cleared by the host's defense?
So are you arguing that the bacteria has been cleared because of the use of the antibiotics, or are you arguing that the bacteria has been cleared because you're able to see that in what you're able to examine in the laboratory?
So in the laboratory, we can treat with antibiotics, and when we treat with antibiotics, the arthritis resolves. So this is not an exact replica of what is seen in patients, because we can treat and follow but in the mice that we don't treat, the level of Borrelia are so low that they're no longer detectable in the joint tissue, but the arthritis is sustained at a high level. So we think that there's something that that is sustaining this arthritis, and it may be what's in patients that allows for a condition where an autoimmune response can develop in the patient joint tissue
Separately. So, but what I'm not clear about is, so you treat the mouse with antibiotics. You're able to resolve the bacteria. At least you're not able to locate any bacteria in the mouse. Yet the arthritic response is still present.
That's right. That's in the mice that we don't treat with antibiotics. So, in the mice that when we don't treat with antibiotics, these mice will sustain severe arthritis for 14 weeks, 30 weeks, without any evidence of Borrelia in the joint tissue. And so we know that there's a tiny bit of Borrelia someplace, but it's not in the joint tissue, yet the arthritis is sustained and the T cells remain activated. So there's something, so there's just enough of an inflammatory trigger that lead to this really robust T cell activation that is sustained for weeks and months in the mice in the absence of the anti-inflammatory.
So one of the thoughts that I had when you and Matt were talking about the interferon and type 1 is, of course, one of the upsides to isolating, a mouse with a particular set of genetics and a strain of Borrelia that is unique, and then combining them, is that, of course, you're able to identify what the response is. And I know you're doing more work on that. But of course, in the wild, what's happening is a tick has a number of different pathogens in, and the tick is spitting a number of different pathogens in, including viruses, right? So your immune response, or actually your hormonal response to the viruses that are being spit into you will be, of course, to respond with the type 1 interferon, right? Which is going to cause inflammation, which then, of course, is going to have the inflammatory response that you've identified when the Borrelia are causing the inflammatory response, which is not an immune response, right? So we have a number of different things happening here when we start to bring the permutations back in. Is we have diversity in the host, the genetics of the host. We have diversity within the strains of the particular germs, and then we have a different combination of germs that are being spit into the into the host, and it's really difficult to even isolate one genetic host with one strain, when in fact in the wild we have a much greater set of permutations, which may be beyond calculation.
I mean, I think that clearly what we do in the lab is reductionist. You've hit it. And the advantage is we can make some conclusions, and we can isolate a single gene or a single protein and look downstream and look at its interactions. But it is a reduction of, certainly, of what's going on in nature and what's going on in people too. So absolutely, there's, we can, the complexity is much, much greater in each of the individual human hosts for Borrelia, and just as you outlined for the wild type with the wild mouse.
So we're seeing patterns develop in the Lyme community that again we've often identified. So, for example, it appears based on at least our podcast and our social media and the interaction that we're having, that there's diversity not only based on like this. There may be diversity based on gender, for example, right? So we saw with COVID that that the female immune system appears to have managed COVID better than the male immune system. And again, I know the data is still being processed, and it appears that perhaps the female immune system may have a different response or capacity to manage Lyme. Are you seeing any diversity in the mouse models with gender?
And we do not. We see that both males and females of C3H and Black 6 strains respond about the same to Borrelia. We see the male mice have maybe a little broader spectrum of arthritis severity, but on average, it's very similar response to the females. And in the mice that we have generated in order to isolate these individual genes on a particular background that we generated in the lab, we're not seeing a difference between males and females. So in our hands, we're not seeing.
Now, why do you think antibiotics don't eradicate and remove symptoms from Lyme in humans, but appear to do so in the mouse models?
I think, that's really hard for me to say. We haven't done, we've done some antibiotic experiments, and occasionally there'll be a mouse that will not have complete resolution, but generally we find pretty good resolution in the mouse. So I think it's just that maybe the control, we can give a higher amount. I can't say.
Dr. Weis, I'm curious about in reading up about all the work that you do, you describe using this murine model of Lyme disease to assess the development of arthritis, following the infection of the spirochete, Borrelia, Burgdorferi. So what is this murine model? Is this something you developed? Can you talk to us more about that model and how you use it in your research?
So by murine, I mean the mouse model of Lyme disease, and it was developed by Steve Barthold. So Steve Barthold's initial observations of C3H mouse developing severe disease, that was the first murine model developed. And so we have based our work on Steve Barthold's initial work, but then we have the IL-10 knockout mouse is one example of a murine model where we get bystander, this overabundant T cell response, bystander T cell activation. The other use of the murine model is the comparison between Black 6 and C3H mice. So it's just another term for a mouse model.
Gotcha. So using the mouse model, the two approaches are those that we discussed, one of them being there's that autoimmune complication with T cells and B cells, one of them being there's not, and it's the interferon creating the inflammation and the symptomology, right?
Yes.
Okay. So, give us some hope here, right? Because most of the listeners on our podcast are suffering from chronic Lyme disease, have tried antibiotics long-term, a wide variety of antibiotics, combination antibiotics. They've tried all kinds of natural herbal therapy. They've tried all kinds of hyperbaric oxygen therapy. They've tried, you name it, anything under the sun to feel better. And they're not, they're still working towards getting themselves into a symptom-free state. And most of the people we interview were living healthy lives until they got diagnosed with Lyme disease and weren't able to recover. So there's definitely a correlation there. So, what word of advice could you give to our listeners, or hope to our listeners, that there are people, really smart people like you, Dr. Weis, that are going to be able to help us understand more detailed, specific things about Lyme to help us get symptom relief and get our quality of life back, in the near future here?
Well, the thing that I would like to do in the last few years of my career is to translate this work to patient populations. And I think, I've had discussions and some pilot studies with samples from people like John Ocott at Johns Hopkins, who has this amazing prospective study where, he's followed patients as at first diagnosis, treated and non-treated, and then can say, oh, look, some patients respond to treatment, some don't. So there, we now have a way to compare and actually look in a very controlled environment of controlled clinical care and have samples from patients at the very earliest diagnosis before treatment to see, are there inherent differences? So these are kind of things I would like to work with, these fabulous clinical studies to try to say, can we see if some of the genes that we're identifying, some of the responses that we're identifying in mice, are these also seen in patients if we have these particular things to look at in a very, in a very fine-tuned mechanism? Can we identify similarities in the patterns of individuals who respond well and those who don't, people who have severe disease and have mild disease? And then these would be novel ways to target and treat. And so we're trying to identify novel targets that we can assess in our animal model easily, and then go to clinical samples, to clinical investigations. Alan Steer is another clinician who I've talked with also to try to say, do we find any similarities? Is there hope that some of these things we're identifying in the mice and we can try in the mice could also have help for the patients in these well-defined patient samples?
Dr. Weis, we really love the way you're using the murine model and the way that you're trying to translate that to the human population. It's just brilliant, and it's really, really exciting. I actually want to ask for your reaction on a different issue, which is, in some cases, what we're seeing is that there is, I guess, the suggestion that what we should be doing is trying to alter the genetics of mice in the wild, that essentially we should be finding ways of altering the genetic composition of mice. And I have to tell you again, I'm going to give you this, my reaction even before I ask for yours, is that really makes me uncomfortable. I'm really concerned about anytime we as a human population make changes to the genetic composition of any animal in the wild. Are you familiar with any of those recommendations and the studies that are being done? And what is your reaction to the belief that perhaps the way to stop Lyme disease from coming to the human population is by altering the genetic composition of mice in the wild?
So I'm not as well versed on those studies as I think you probably are. I think these are they being done on Martha's Vineyard or in Nantucket, and they're mice expressing an antibody against Borrelia ospae, right? So I don't, I think that it's a situation of, in theory, the idea is that you would spread through the entire mouse population, mice that would be resistant to Borrelia. And of course, the caveats are that we know there's so many different, that there's ability to select for Borrelia expressing different outer surface antigens that might bypass the particular antibody being expressed by that mouse, and also that other small animals can serve as reservoirs for Borrelia burgdorferi. So I think I share your concern. I guess my concerns are, will this be effective in the long term, or will you end up with different Borrelia or other animal reservoirs that become more important than the mouse? But I think that, in theory, the idea of trying to control the mouse population and control the reservoir host for Borrelia burgdorferi makes a lot of sense in these refined communities. I don't know that there's a lot of, I don't have a lot of fear that the mice are going to somehow alter or make a super mouse that's going to take over the population. I guess my concern is what is the long-term effectiveness of making a mouse resistant to a particular isolate or few isolates of Borrelia burgdorferi when there are so many other potential isolates that could then rebound.
But it really is a slippery slope. For example, you're genetically altering mice in your lab for the purposes of isolating a particular response, but you're not releasing that population of mice that are being genetically altered into the wild to alter the mouse population. And I think it is a slippery slope. I think once we start to alter genetic code, and once we start to release that into the wild, we don't know what the impact is going to be. And I think once we go down the slippery slope, we may be creating Frankenmouse. And I know you seem to be less concerned about that than I am, but I think it is a slippery slope. And I think it's in a carefully controlled environment like yours, it makes sense, and it's beneficial. I think when we're trying to begin to genetically alter animals in the wild, I think it's a slippery slope that may send us in a place where we may be getting outcomes that are much worse than those that we're trying to protect against.
Yeah, I think that there are a lot of very strong feelings about this. As I say, I'm really not an expert on, I know there have been many, I think that there have been town halls and things to discuss this.
And I think it's more of an ethical question than anything else, but because we were having the conversation about murine modeling, because it's such a large part of your work, I was just wondering what your reaction would be. But I understand it's off topic. So why don't you, as sort of a way of winding down this podcast, answer the question we ask everyone on our podcast, which is, if God forbid one of your daughters came into your room right after this podcast and they found a tick biting them, what would you recommend they do so they wouldn't go on a chronic Lyme disease journey?
Well, since one of them's an MD and one's and her sister's a PhD, depends on what one of them would be telling me exactly what to do, and the other one would be calling her sister. So, but at the University of Utah, I mean, I do get phone calls a lot, emails and phone calls with people who are very concerned about relatives that are coming to visit, or they've been in an endemic area, or they've been told they have Lyme disease. And we have a wonderful infectious disease department at the University of Utah, a big global medicine group. So even though we don't have very many cases of Lyme disease in Utah, we do have people who vacation elsewhere, who come here, or and so there are several physicians in the infectious disease division who I know are experts and very knowledgeable. And I always direct patients to see those physicians at the University of Utah. And I say, you can use my name, you can email this person, and they will definitely try to get them in. They're very experienced. We have great rheumatologists, rheumatology community too. And that's what I do at the living in Salt Lake City, is I try to connect people with really knowledgeable physicians at the University of Utah or other local hospitals.
Professor Weis, we can't thank you enough for taking time out of your really busy schedule, not only getting up really early, but taking time out of your busy schedule to do this podcast. And we thank you for all of the great work that you're doing. And we really can't wait to see, as your career progresses, we can't wait to see how you bring your work into the clinical setting and hopefully help us resolve more and more of the suffering that's being caused by Lyme disease. So thank you so much.
Thank you. It's really, it's a pleasure. And I wish everyone the best. I hope that you both do well. And I hope all of the patients that are listening, I'm very hopeful for all of you. So
thank you.
It's a privilege to get to speak to you. Thank you.
Thank you for listening to our Tick Bootcamp interview with our guest, Dr. Janis Weis. To our listeners, we have a call to action. First, if you'd like to learn more about Dr. Weis, please check out the University of Utah's faculty webpage at medicine.utah.edu/faculty. Second, if you've enjoyed this episode of our Tick Bootcamp podcast, please share it with your friends on social media. Third, Tick Bootcamp has created a Tick Bite blueprint that has been inspired by the information that has been shared with us by past podcast guests. We urge you to visit our website at tickbootcamp.com/bite to view our blueprint. Fourth, don't forget to subscribe to this podcast on Apple Podcasts, Google Podcasts, or Spotify to get your automatic episode updates of our Tick Bootcamp podcast. Please take a minute to leave us an honest review on your podcast platform of choice. And finally, if you'd like to search our podcast library of almost 300 episodes, subscribe to our email list, or share feedback with us, please visit our website at tickbootcamp.com. Thank you, as always, for listening.